videos left, guys. Let's get through this. Sign in and comment down below if you have any questions. DNA structure, right? So we know that DNA structure is a double helix, and through Hershey's experiment, we also know that A and T pair together. Why do A and T pair together? Well, that's because of the fact that there's pyrimidines and purines, so the number of rings and the number of hydrogen bonds, which we'll go over in the next slide. But this slide serves really nicely to kind of remind us that what a nucleotide is made up of, right? You have your phosphate group, your sugar, so in DNA deoxyribose, and then your nitrogenous base, right, which can be adenine, tyrosine, guanine, or cytosine. Yeah, so another thing to point out is that the strands are labeled in direction according to the sugars, the ribose's carbon, so from 5' to 3', and the strand is anti-parallel, right? So although one strand goes from 5' to 3', then the other strand will then go to 3' to 5'. So DNA is anti-parallel because of this, and it is complementary because of guanine and cytosine being paired up and tyrosine and adenine being paired up. So it is a anti-parallel complementary double helix. So like I said, about the nitrogenous bases, you have your four nitrogenous bases: guanine, cytosine, adenine, and tyrosine, right? And I like to use this mnemonic to remember which ones are purines and which ones are pyrimidines, and also the pairings of them. So it's called GCAT, puppy, puppy, right? So your guanine and your adenine are your purines, which means they have 2 rings, as you can see right here, and your cytosine and your tyrosine are pyrimidines, which means they have 1 ring. And the reason that 1 ring and 2 ring has to line up is because if you had 1 and 1 ring, it would be too short of a distance to have a linear anti-parallel strand. And if you had two membered nitrogenous bases, then you would have 4 total, which would be 2 wide. Sorry, that took a lot to articulate. And then talking about the hydrogen bonds, right? Guanine and cytosine are paired up instead of guanine and tyrosine, because guanine and cytosine have the ability to make 3 hydrogen bonds, whereas adenine and tyrosine are 2. And a question that's asked about the GC content a lot refers to organisms that survive or thrive in high heat environments, and you can conclude that or infer that a organism that survives in a high heat environment would have a high GC content, because a triple 3 hydrogen bonds takes more energy to break than 2 hydrogen bonds, which means that the high heat environment would not denature the DNA as much as it would if it was higher in AT concentration. So if you remember GCAT puppy, you'll remember which ones are paired together, or the bonds between each of the nitrogenous bases, and the amount of rings that they have as well. So this is kind of helpful. Okay, so the last thing about structure that was yet to be identified had to do with replication, right? So how does replication occur? Are you getting 2 strands like that look like this, so an original strand and a new strand, so this is the semi-conservative conclusion, or are you getting a strand that is completely new, and then this would be your old strand, so it would look like this and then this? Or are you getting a dispersive one where there's segments of each? And this experiment by Meselson Stahl served to prove that. So what happened was they labeled DNA based on their weight. So they used nitrogen isotopes that had different weights, so nitrogen 15 and nitrogen 14. And when those were suspended those samples were suspended in a test tube, you could identify uh, what DNA was present based on the weight. So something that would be lighter would be higher up in the test tube, and something that would be heavier would be lower in the test tube. So what happened was, originally you start with your nitrogen 15, right? You're growing your bacteria in your nitrogen 15, and you have 100% heavy DNA, right? Then what happens is, generation 2, you now take your bacteria that was grown in nitrogen 15 and you put it into a nitrogen 14 solution. So now any new strand that is going to be made is going to have nitrogen 14 and thus be lighter. And what do you get from that? You get a middle band, right? So that indicates that this one double helix was not pure nitrogen 15 and was not pure nitrogen 14. So that's why you get a strand in the middle. So the next generation that was still grown in nitrogen 14, you see a band at the top, which is purely new, made of purely new isotope, nitrogen isotope, which is indicative of like these two double strands, right? These two helixes that are completely made of new DNA. And then you still have your one original strand and your new strand right here. So this then proves a semi-conservative approach that you get, that with each replication you get one strand in the double helix that is old and was the template, and then a new strand, right? And if this was actually the conservative approach, you would get a strand of 100% old and a strand of 100% new in this generation. And if it was dispersive, then you would just get a middle band throughout, right? Because there's nothing distinguishing between each generation. Okay, so in DNA replication so this proves that DNA replication was semi-conservative, right? So then this is a nice summary of how DNA replication happens in general. And DNA replication happens in interphase, right? As the cell is preparing to divide, you need to replicate your DNA. So there's 4 major steps that you need to keep track of: unwinding, initiation, elongation, and termination. So unwinding refers to the fact that you have this double helix that's kind of wound up, and you need to expose your nitrogenous bases for anything to even be replicated, right? So unwinding occurs, and helicase is the enzyme that is responsible for unwinding. And as these two things unwind, you cause a lot of tension in your strands. And if you look back in your lectures or even Google this, the example that is shown is a rope that is being pulled apart. At the end of it, you're going to get these coils. So then the enzyme that relieves that tension caused by the coils is called topoisomerase, and topoisomerase cuts the coil and unwinds it and joins it again to relieve tension. So we've gone over 2 enzymes. You have your helicase and your topoisomerase, and then you have your single-strand binding protein, and this protein serves to hold the replication bubble open, so to prevent the DNA from winding up again as this process is going on. Next is initiation. Initiation happens with a primer and a primase. So you get a primer come and attach and a primase as well. And this will be the starting site for DNA polymerase 3 to come and bind to start elongation, which is the adding of nitrogenous bases. And up here is a really good example. You see that the origin of replication is in the middle of the bubble, so it's important to realize that. And it's also important to realize that you have a leading strand and a lagging strand, right? So here you can see that DNA is produced 5'. DNA is produced 5' to 3', right? So regardless of which way you're going, you have to start at the origin. So here you kind of have to work backwards. It's called a lagging strand. It takes a lot longer to complete as well. So here you have the same enzyme causing elongation, right? But what's different here is that you have DNA polymerase 1 come and replace these primers, because you're going to have a lot more primers in your lagging strand, as you can see right here. Here you can do a continuous elongation, but here, since you're kind of working backwards, right, because this is the origin, you have to go like this, then like this, then like this. You have more primers here, so those primers need to be replaced, so DNA polymerase comes and replaces them. And then the trusty DNA ligase is responsible for gluing them back together. So ligate means to join again, so DNA ligase is responsible for gluing the lagging strand back together. So this kind of overviews DNA replication as a whole. There's a lot of great videos on the internet that probably explains this process a lot better than I do, but I can only do my best. So we talked about elongation, and then termination happens when everything is finished and you have your two separate strands. Okay, so next we're going to talk about chromatin cracking, right? So we're very familiar with this from our overview of mitosis and meiosis. This is our chromosome that's condensed, and we're also familiar with this double helix, right? And we know both are DNA, but we don't necessarily know what happens in condensing before this point to get from a double helix to a chromosome. So this kind of outlines what happens, right? So you have your double helix, and your double helix is this blue strand that is then just represented by this line, okay? And you have your histones, and what happens is your long strand, your long double helix wraps around these histones, which are proteins, okay? And it creates like this beaded necklace, right? And then these beads further stack up against each other, and then this long strand, this purple strand, it then goes through scaffolding right here, so it kind of folds in like this. Then these long scaffolded chains will then condense into a chromosome. So as you can see, it's there's a lot that goes on between a chromosome and the double helix in terms of condensing to really optimize or to create an efficient division process, right? If you had this DNA unwound and floating about, it would not be as orderly than having these nice compact chromosomes just pull apart in anaphase. So I'm gonna again cut the video off here. Let me know if you have any questions and sign in. And the next video we'll talk about the central dogma and specifically focus on translation and protein synthesis. So far we've talked about DNA replication, we've talked about transcription as well, or we will talk about transcription and highlight protein synthesis as well. So I'll see you in the next video.